My name is Titia de Lange. I work at the Rockefeller University in New York, and today I'm going to talk to you about telomeres, their structure, their function, and their role in human disease, and specifically in the development of cancer in the human system and a set of diseases that are caused by telomere dysfunction. So telomeres are the ends of chromosomes. They're elements that protect chromosome ends, and they're defined by a specific sequence that is highlighted here in this metaphase spread of human chromosomes by a probe that can detect that sequence. So let's first examine the sequence of telomeres in detail. Telomeres are made up of a very long array of a simple repeat. The sequence is TTAGGG. And this sequence is repeated on and on along the telomeric DNA, which can be very long. In human cells, it can be as long as 10 kb. In mouse cells, it can be as long as 50 kb. Many thousands of copies of this simple TTAGGG repeat. And then at the very end of the telomere, there is a special structure. There is a 3' protrusion, that you can see here, of the single-stranded G-rich strand of the telomere that sticks out. So the 5' end strand is shorter than the 3' strand. And this is quite long. It's 50 to 300 nucleotides. And this structure is very important for telomere function. Telomeres are needed at the chromosome end to solve one critical problem that cells have. And I refer to this problem as the end protection problem. The basis of this problem is the following. Cells have a very vigilant way to identify and respond to breaks in their DNA. So if there is a break somewhere in the chromosome, the cell will recognize this break and activate what is called the DNA damage response. The DNA damage response is guided by two kinases, the ATM and the ATR kinase, which each can be activated at this DNA end, and then through a series of phosphorylation events, guide the cell to stop the cell division, and they orchestrate DNA repair. Now, if you look at a telomere, of course, it looks very much like a double-stranded break. But this structure should not activate the DNA damage response, because if the ATM and ATR kinase became active at our natural chromosome ends, the cells would never divide. And cells would try to repair the ends of their chromosomes, which would lead to genome instability, as we will see later in the talk. So the main function of telomeres, the essential function of telomeres, is to make sure that they are ignored by the DNA damage response. So how do telomeres do this? Well, the telomeric DNA itself doesn't do very much. It's not good at repressing the DNA damage response. It needs to function by recruiting onto the telomeric DNA a protein complex that we've called Shelterin, because it shelters the chromosome end. Shelterin binds all along the telomeric DNA through the action of two proteins in the complex, TRF1 and TRF2. These proteins bind to the double-stranded TTAGG repeats. They then help to recruit into the telomeric complex a protein called POT1, the red guy that you see here. And POT1 has the ability to bind to the single-stranded telomeric DNA. And then there are other proteins in the complex, TPP1, TIN2, RAP1, that hold the complex together. Shelterin is very abundant. There are many hundreds of copies of Shelterin at every telomere, and every cell expresses these proteins. So in every cell of our body, the telomeres are covered by this protein. The complex is highly conserved in mammals, but it's not found in some other organisms like yeast or plants, or it's different. But because it is conserved in mammals, we have been able to use mouse model systems, mouse cells, to study the complex in detail. Telomeres also contain other proteins that I never put in my slides because it's too hard to draw. They contain nucleosomal chromatin, so the telomeric DNA is wrapped up into nucleosomes. You can understand more about nucleosomes if you listen to David Ellis's talk. And telomeres contain Shelterin accessory proteins. These are proteins Shelterin recruits onto the telomere in order to fulfill its function. So Shelterin blocks these two major signal transducing pathways that respond to double-stranded breaks. It has to block the ATM kinase pathway and the ATR kinase pathway. So how does this happen? So the ATM kinase pathway is activated by DNA ends. The DNA end is bound by a terminal protein complex, MRN. This complex stably associates with the DNA end at the DNA break, and then it recruits onto the DNA end the ATM kinase. And in ways we don't fully understand, the ATM kinase then becomes activated. Once the ATM kinase is active, it phosphorylates proteins in the vicinity of the break in a very specific way. And one of the proteins that's a target of ATM is a histone variant, H2AX. This histone variant is found throughout the chromatin in the nucleosomes. So whenever a break occurs, there is some H2AX in the vicinity. The phosphorylated form of H2AX now functions as a platform for the recruitment of proteins that are going to make up the DNA damage foci. The first one is MDC1, which initiates an ubiquitination cascade that leads to the recruitment of other proteins, including this protein here, 53BP1, which is a protein we'll use as a marker for DNA damage in this talk. These DNA damage foci help to both amplify the ATM kinase signal and to guide the repair at the DNA break in appropriate ways, depending on the cell cycle stage in which the break occurs. So this is the repair part of the ATM kinase. But it also impinges on cell cycle progression. The ATM kinase will phosphorylate an effector kinase called CHEK2. CHEK2 then phosphorylates other proteins, including p53 and the CDC25 phosphatase, and together these phosphorylation events lead to the inactivation of the cyclin-dependent kinases that normally will help cells to progress through the cell cycle. So this is the basis of the cell cycle arrest. Obviously, none of this should happen at telomeres, even though the telomeres look very much like an MRN binding site. Shelterin needs to shut down this program. How does it do this? Our current model is that Shelterin manages to block the ATM kinase signaling pathway at natural chromosome ends by remodeling the telomere into what we call a T-loop. We discovered, together with Jack Griffith, who works at the University of North Carolina, Chapel Hill, who's an expert electron microscopist, that telomeres, the telomeric DNA from human, and in this case, what is shown here, from mouse cells, is not linear, but ends in a lariat structure. And detailed analysis of these structures, these are protein-free DNA structures, suggested to us that telomeres use that single-stranded overhang that I talked about earlier to make this T-loop structure. The single-stranded overhang strand invades into the double-stranded part of the telomere, and it's Shelterin that does this job. And in doing so, it basically hides the chromosome end from any activity that could potentially load on the chromosome end. So, because of this, the end is hidden from the DNA damage response. That is, while at a broken end, MRN can bind and activate the ATM signaling cascade. At the telomeres, MRN cannot find a binding site, and therefore the ATM kinase doesn't become activated. So this is how Shelterin deals with the ATM threat at chromosome ends. How about the other transducer, the ATR kinase? The ATR kinase is dedicated to finding DNA lesions in the genome once they have attained single-stranded DNA. So a double-stranded break can be digested by a nuclease, leading to a long single-stranded overhang that can be recognized by the ATR kinase. And it does it through the agency of a very abundant single-stranded DNA binding protein, RPA. RPA is all through the cell, will accumulate at single-stranded DNA, and it acts in conjunction with a ring-shaped molecule, 911. Once these proteins are loaded at a broken DNA end that has been resected, they recruit and activate the ATR kinase through a number of interacting factors. And now this active ATR kinase at the site of damage will do pretty much the same thing that ATM does. It phosphorylates H2AX, and H2AX then recruits the proteins that form the DNA damage foci. And ATR, like ATM, phosphorylates a downstream effector kinase that will impinge on the cell cycle progression through the phosphorylation and destruction or inactivation of a CDC25 phosphatase. How do telomeres prevent this kinase from mistakenly recognizing natural chromosome ends as sites of DNA damage? After all, the telomere contains the exact structure that activates the ATR kinase. It has single-stranded DNA that extends 3'. It has a 5' double-stranded, single-stranded transition. It has what it takes to activate the ATR kinase. Now, Shelterin has this wonderful trick in that it contains POT1. POT1 is a single-stranded binding protein, and by being at the telomere and covering the whole single-stranded part of the telomere, it prevents the loading of RPA onto the telomeric DNA. So there's not just one POT1 sitting there, but in fact, the whole single-stranded part of the telomere is covered by POT1, and because of that, RPA simply can't bind and therefore will not activate the ATR kinase. So now we've seen how Shelterin has two main tricks by which it prevents the activation of these DNA damage response pathways at telomeres. So one problem solved, but in fact, there is a second problem with telomeres, and that is the problem that telomeres shorten with cell divisions. So in this southern blot, an old-fashioned technique that my lab still proudly uses, we look at the telomeres of primary human cells that are undergoing numerous cell divisions. They're grown in culture. And then what we do is we cut the telomeric DNA loose from the rest of the chromosome, separate it based on size on a gel, and then we detect the telomeres with a probe that can anneal to the telomeric DNA. And you see that human chromosome contain telomeres that are very heterogeneously sized, but as you look over the population doublings, this smear of telomeric DNA becomes shorter. There are two bands in this blot that don't become shorter, and they serve as a wonderful control, and they are chromosome internal sites where there is a little bit of telomeric DNA. Why does this happen? Well, there are two processes that give rise to this shortening. The first is the well-known end-replication problem. It's a problem associated with DNA polymerases that try to replicate a linear chromosome. DNA polymerases always synthesize DNA from 5' to 3'. So they use a 3' hydroxyl to add a nucleotide and another one and another one. So they move in the 3' direction. So when telomeres replicate from an origin somewhere in the chromosome, moving the replication fork, moving out through the telomere, one polymerase has no problem. That's the polymerase that does leading strand synthesis, and it just zooms out to the end. But the other strand cannot be replicated in this direction. It has to be replicated in the other direction. And that is what is called lagging strand synthesis. And because the polymerase goes from 5' to 3', it needs to do it in little steps, and it needs a 3' hydroxyl to start each of these little steps. And in this lagging synthesis, it's the RNA primers that are laid down by primase, part of the replication machinery, that allow lagging strand synthesis to synthesize DNA in this direction. There you go. And before you know it, you have almost replicated the whole strand. The RNA primers are now removed, and you can fill in this little gap here. Aha, but here is the problem. At the very end of the telomere, there is a gap that can't be filled in because this gap has a 5' end, and there's no polymerase that can go in this way. So this is a fundamental problem, first discussed by Jim Watson with DNA replication of linear chromosomes, and it leads to gradual and progressive sequence loss. But in human cells and in mouse cells, there is an additional problem, because the product of this DNA synthesis scheme has not recreated the structure that we need at telomeres. There's no long 3' overhang. So what Shelterin does, it recruits onto the telomere nucleases that are going to eat in the 5' ends, both 5' ends, and digest away the end to make this long overhang. And there you go. Now we have reconstituted the telomere in its original structure, but in doing so, the nucleases have removed quite a lot of DNA from each chromosome end that was newly replicated. So together, the end replication problem and this chewing in of the telomere to make the correct structure leads to the rapid loss of telomeric DNA that you see in this experiment. Now, there are consequences. If you lose telomeric DNA, eventually 1 or 2 telomeres of the heterogeneously sized population of telomeres will lose its ability to repress the ATM and ATR kinases, and then cells will arrest. This is generally referred to as the Hayflick limit. Len Hayflick first observed this when he was culturing primary human cells, that after 50 population doublings or so, the cells would go in what is called senescence. Senescence is a terminal state for cells. They can't get out of it. They stop dividing, and they show a lot of phenotypic alterations, including a marker that we stained blue here in this particular experiment. So what is happening here is that as the cells are dividing and their telomeres get shorter and shorter, for a long time, there is no consequences, but once 1 or 2 telomeres have lost their ability to repress the ATM and ATR kinases, they will signal and activate a program that leads to permanent cell cycle arrest, senescence, or in some cell types, it can lead to apoptosis. So this is a problem because our telomeres shorten, which has consequences. How can we live? How can we breed? How can we produce progeny if this goes on in our germline? Well, it actually does not go on in our germline. In our germline, there is an enzyme, telomerase, that solves this problem and adds sequences back onto the telomere. Telomerase is a reverse transcriptase. It was discovered by Carol Greider and Liz Blackburn, and they received a Nobel Prize for this discovery together with Jack Szostak. Telomerase is a reverse transcriptase, so it copies an RNA into DNA. So this big orange thing here, that's the RT, the reverse transcriptase. And then you see the RNA there. And this RNA contains in it a very short sequence that encodes the telomeric DNA. And then it has some additional factors that help in biogenesis that I colored in blue here. So Shelterin is recruited to the telomere by, telomerase is recruited to the telomere by Shelterin, and then it can align its RNA template sequence with the 3' end of the chromosome. And will then use the 3' end of the chromosome to add more telomeric repeats, like so. There is one repeat. This is only 6 nucleotides. But it can translocate on the DNA and add more and more and more repeats until it pushes me out of the slide. So telomerase can solve the loss of telomeric DNA by making up for it, by adding DNA back of the exact right sequence onto the chromosome end. And it does so in stem cells and in the germline. So both in the germline and some stem cells compartments, telomerase is expressed, and it maintains the telomeres intact. However, during normal development of our cells, of the somatic cells, the cells that make up the tissues in our body, telomerase is silenced. Somatic cells do not express the third component of the enzyme. The third promoter is switched off in ways that aren't fully understood. And as a consequence, all our cells show this telomere shortening program that I explained to you. Now, as I said, telomere shortening will erect a proliferative barrier. No matter what cell type it happens in, at some point, those cells will lose telomeric DNA to an extent where they will activate the ATM and ATR kinases, and that's the end of that cell lineage. So why? Why do we have a system in our cells, in our tissues, not in the germline, not in the stem cells, in our tissues, that eventually will block the cells from proliferating? Well, it's generally believed that this has evolved as a tumor suppressor pathway. If you think about it, if during the formation of breast cancer early on, an epithelial cell, a mammary gland, experiences a mutation in a gene that is involved in cell signaling that creates a situation in which the cell starts proliferating inappropriately, that cell clone will go through a number of cell divisions, but since it doesn't contain telomerase, it will stop dividing after a number of PDs. The DNA damage response will be activated. P53 and RB will start operating, and you get induction of apoptosis or senescence. This is thought to be a very powerful tumor suppressor pathway that can stop tumorigenesis in breast cancer in a very early stage, in a hyperplasia stage. However, like all tumor suppressor pathways, occasionally and unfortunately, it fails. And if you think about it, one way in which this pathway can fail in trying to stop incipient cancer cells from growing out is through the loss of the two tumor suppressor pathways, P53 and RB, which are frequently lost if you look in clinically detectable cancer. And now something very different happens. The cells continue to divide without telomerase and continue to lose telomeric DNA. And you create a situation in which the cell now contains not just one or two, but many dysfunctional telomeres. And this state is referred to as telomere crisis. And in telomere crisis, what occurs is that the dysfunctional telomeres start fusing together because they've become so abundant that they can find each other. And it's basic DNA repair that executes this job. It's non-homologous end joining that does this. This is a repair pathway that normally gets activated at broken DNA ends. It's a ligation reaction. It just takes two DNA ends and fuses it together no matter what the sequence is. And this very same reaction is repressed at telomeres, like the ATM and ATR kinase pathways, by Shelterin. So normally at telomeres, there is no non-homologous end joining. But when cells have experienced ongoing telomere shortening, leading to a state where now not just one, but many telomeres contain too little Shelterin because they're too short, and therefore do not block DNA repair, non-homologous end joining can occur at the chromosome end. So what happens under those conditions is that the non-homologous end-joining pathway finds two telomeres, because there are now many, and they can find each other in the nucleus and ligates them together. And this is a disaster, because the chromosome that has been formed by this reaction now has two centromeres. And centromeres function to allow chromosomes to segregate in mitosis by attaching to the spindle apparatus. But the dicentric chromosome that has two centromeres can misattach during mitosis, as shown here, and lead to a problem in cell division. So if you have a cell with dicentric chromosomes like this one that we filmed in our lab, a movie made by John Masijowski in my lab, you can see the cell struggling with this connection between the two chromosome clusters. It's been studied in detail what happens to these events, and we now understand that the telomere crisis situation with their many dysfunctional telomeres and the problems that dicentrics create can lead to a whole array of genome alterations. First of all, we know that translocations can result from this situation. This was shown by Ron DePino, modeling in the mouse. We know that regional amplification can occur as a consequence of telomere crisis. We've recently shown that two new phenomena in cancer genomes, chromothripsis and kinases, can arise in telomere crisis. Chromothripsis is a massive, sudden shattering of a whole part of a chromosome, and then the pieces are put back together in the wrong orientation and order. Kinases is a storm of mutations that occur at the breakpoints of these chromothriptic events. These two can be due to telomere crisis. And finally, we found that the ongoing DNA damage signaling originating from the many dysfunctional telomeres in crisis, even though the cells don't arrest, they still experience the ATM and ATR signaling. This can lead to cells making a mistake in the cell cycle, where they don't synthesize DNA and then divide. They synthesize DNA, don't divide, synthesize DNA again, and then divide. And this leads to an endo-reduplication, a doubling of the genome, and this is called tetraploidization. This is a phenomenon that's quite general in cancer. Many cancer genomes show evidence of tetraploidization, and tetraploidization has been shown to promote tumorigenesis. So just as an example of these kinds of alterations, I show here a metaphase karyotype of a cancer cell line. This cancer cell line shows clear evidence of tetraploidization. This is a sky analysis in which people have colored each chromosome with a different color and then aligned them. They don't fall out of the cell in metaphase like this. And you can see that, for instance, chromosome 2 here in red, it's present in 4 copies, as is chromosome 3, as is chromosome 4. This presence of 4 copies of one chromosome indicates that this cancer originally went through a tetraploidization event. And after that, it loses chromosomes readily because tetraploid cells are quite unstable. In this metaphase karyotype, you can also see that there are many translocations in these cells where colors from one chromosome are now linked to colors of another chromosome. So this kind of genome instability can be ascribed to telomere crisis. That's what we have proposed, and we're studying this further. It's not easy to study because telomere crisis, again, like the tumor suppression, occurs very early in tumorigenesis. Using the example of breast cancer, it's been shown that telomere crisis occurs in DCIS, ductal carcinoma in situ, in the earliest stage of breast cancer that is actually quite unlikely to go on to be a lethal disease. So in that state, The telomeres have run out of their protective ability. Cells have lost the tumor suppressor pathways. There's no telomerase yet, and they show enormous genome instability. But these cells are not going to grow out into a tumor. Cells in telomere crisis are deeply unhappy. They struggle to divide, and they will only give rise to a frank tumor if telomerase becomes activated. And it's in telomere crisis that there's very strong selection for reactivation of this enzyme. Telomerase is needed to resynthesize the telomeres, to heal them. This will lead to a stable genome. Cells now can divide appropriately, and they will come out with this heavily rearranged aneuploid genome that can promote tumorigenesis. And indeed, telomerase is seen to be activated in greater than 85% of cancers. The other cancers have a different way to maintain telomeres. And in some of the cancers, telomerase is activated by mutating the promoter of telomerase, of the third component of telomerase, breaking through the silencing program that normally is instituted during development. So what I discussed with you is quite a complex dual role of telomeres in cancer. It starts out with programmed telomere shortening in our normal somatic cells. This is a good thing because it will create a proliferative barrier for the cells, and it will stop tumorigenesis. But in rare cases, when cells break through this, the same telomere shortening program can lead to genome instability, which of course could lead to oncogenic changes and promote tumorigenesis. And then in the end, telomerase needs to be activated for the cells to come out of this and form a clinically relevant tumor. Now, this is one aspect of telomere biology in human disease. I want to end by talking about a second disease-relevant aspect of telomeres. And those are the inherited diseases that are caused by telomere dysfunction. It's now clear that there is a large set of diseases that are inherited that are due to short telomeres. In some cases, there are developmental abnormalities that play out in utero. In the disease that first brought this principle to light, this keratosis congenita, the short telomeres give rise to bone marrow failure early in life, very dramatic. But in adults, there are now also cases of aplastic anemia, idiopathic lung fibrosis, and liver cirrhosis that are ascribed to a defect in telomere maintenance. So these diseases are clearly familiar, very well-defined, large family pedigrees, and they're always due to telomeres being too short. So if you look at telomere length in the human population, telomeres shorten a bit with age. It's not much, but they do shorten a bit. What most people don't realize is that there is enormous variation in telomere length. So at any given age, the telomeres can be quite long or short depending on the individual. But even in the face of this variation in telomere length, it's very clear that the people having these diseases, colored here, have extremely short telomeres. And these extremely short telomeres, which they get at birth, of course will lead to premature cell cycle arrest or cell death in the tissues, just in the same way I described this to you for primary human cells grown in culture. So the diseases that I described are due to mutations in two pathways. One is the telomerase pathway. You see that almost every component of telomerase is mutated in one or another of these families. So the third component, the reverse transcriptase, the TER component, the RNA of telomerase, NOP10, NHP2, discarin, the blue guys that are needed for biogenesis with telomerase, TCAP, a trafficking protein, and PORN, a protein needed for maturation of the telomerase RNA. You see mutations in these different genes, and in each case, the mutations do not fully remove telomerase, but it lowers the telomerase level so that the telomeres are not fully maintained in the germline and in the stem cells in the patients. The other pathway that's affected in these diseases is Shelterin. You see mutations in TIN2, POT1, and TPP1, and they're likely to affect the way telomerase is recruited onto the telomere, again leading to deficiency in telomere maintenance. And then finally, there are some proteins that are Shelterin-associated factors whose function in the telomerase pathway is not yet fully understood. So let me finish by reiterating the logic of telomeres. This is how telomeres work. Telomerase, beautiful reverse transcriptase, goes to the chromosome end and it maintains the TTA-GTG repeats. These repeats don't do much by themselves. They need to recruit this protein complex Shelterin in order to protect the telomere. Shelterin modifies the structure of the telomere into a T-loop, represses the DNA damage response also through its POT1 component, and makes sure that telomeres are not seen as what they actually are at the end of the chromosome. So I've talked about work done by a very large number of labs. I haven't had the opportunity to mention everybody. That's not the purpose of this kind of lecture, but please, if you're interested, read the reviews and find the right references. Our work is done at the Rockefeller University, beautiful campus on the Upper East Side of Manhattan, where I've been for 26 years, and currently we are supported by a number of funding agencies for which I am very grateful.